So we'll review the two chapters that are in Unit 3, which is Chapter 29, and then Enzymes is Chapter 30. So we have kind of a narrow focus on this unit, so it's pretty easy, so to speak. So here's one way to show an amino acid with its stereochemistry using the Fisher projection. And they all are, or I should say the average amino acid is L. Glycine, one of the 20 amino acids that you should be familiar with that are found in proteins, has no chiral center because these two are the same. And then, as we talked about in the chapter on carboxylic acids, at physiological pH, which is somewhere around 7.4, 7.3, most carboxylic acids are ionized. And in the chapter on amines, we see that, yeah, most amines are also ionized at physiological pH. So under these conditions, amino acids are Zwitterions. They have both a positive and a negative charge. So two of them would be strongly attracted to each other by ionic attractions. And because all amino acids share this same organization right here, you should know how to draw them. And as far as R goes, you should know at least glycine, which is the simplest one, it's just a hydrogen. Alanine, which is the next simplest one, which is a methyl group. Phenylalanine, which is simply a phenyl group on the methyl group. Cysteine is a thiol. Lysine is primary amine, and aspartic acid is carboxylic acid. And then you should be able to classify all of them, no matter what you're given, as either neutral nonpolar, neutral polar, acidic, or basic. And in terms of electrophoresis and isoelectric point, yeah, you should be able to draw them at physiological pH. So this is physiological pH, right? Both the amino group and the carboxylic acid group are ionized. If you get to one where you have an acidic R group, okay, well, how is that going to work? Well, the PI is 2.77 for this particular amino acid. And what that means is that at the PI, there is no net charge. So if I'm looking at this right now, there's no net charge. If I plop it in right here, where there's a very, very low pH, there's a lot of hydrogen ions around, I'm going to protonate everything that can be protonated, including the two carboxylic acids. So now, at this pH, there's a net charge of +1. It's going to be repelled by that electrode, attracted to that electrode. It's going to migrate in the electrical field. It's going to electrophorese through this material until it gets to the PI. If I put it way over here, where you have mostly hydroxide ions, you're going to pull hydrogens off of everything. And when you do that, you have a net -2 charge. So it's going to be repelled by this, attracted by the positive electrode. It's going to migrate this way until it hits right here. That's where it has no net charge. So it congregates here, it focuses over here, and we can precipitate it, and we can separate from other amino acids. So here's a basic amino acid. This is lysine, so it's a primary amine. Right now, it is at, the way it's shown, has no net charge. Okay, so this is what it would look like at pH 9.74. If I were to take this and put it into an acidic pH, And that would be protonated, that would be protonated, that would be protonated, and it would have a net charge of +2. So it would migrate that way. If I had it at a really high pH, I would take protons off of everything. So I would take the proton off of here, and this would have a net charge of -1. So it would migrate that way until it got to its pi, which is somewhere around here. Okay. So while we're at it, somebody had sent me an email. I hope this is the screenshot. Yeah. So here's the screenshot. This is from the review. Okay. So looking at this problem here, she's asking, or number 4, is the reason that it would migrate towards the negative electrode is because it's ionized. Yeah. So here is, let's see, there's a no, and then here, okay, so this has a charge of +1 at neutral pH, right? So at neutral pH, it has a charge of +1. Is it going to migrate in an electrical field? Yes. It's not going to remain stationary. It's not going to go towards the positive electrode because it itself is positive. So it's going to migrate towards the negative electrode. Hopefully that makes sense. No? Somebody talking? I can't. Anyway. Then number 4 is the reason that it will migrate toward, oh wait, this one. So what does classification by composition means? Well, it's either going to be a simple protein or it's going to be a conjugated protein. Simple protein is only made up of amino acids. Conjugated protein has something besides amino acids. So hemoglobin has a heme, has an iron. So therefore, it's a conjugated protein. Okay. So I'm never going to ask you for the structure of a hypoglycin or something like that, but you should know that there are amino acids out there that are toxic. And then levels of protein structure. They're all tied together, which is how the base sequence in the DNA basically gets to the point of prescribing its function. Because a certain primary structure will dictate whether it's going to be alpha, beta, or random. And this structure right here, how it folds together, will determine whether it's going to be fibrous or globular. And then, if it's a monomer, doesn't matter whether it's fibrous or globular, that determines, is it going to be a monomer or an oligomer? So what you need to know how to do is take 2 amino acids and put them together in a certain way. So you need to be able to draw the peptide bond. And then this whole thing about the pKa, I was really just trying to demonstrate to you that this is going to have a different pKa than this one. So in other words, alanoserine is different than serinoalanine. So then we talked about sequence homology. So here's insulin. Here's the processing of insulin. So this is what's coming from the gene. So the gene well, we'll see later on, it may or may not have introns in it, but the gene codes for this messenger RNA, which codes for this protein, but that's not the protein that we see cruising around in the blood. It has to be processed. So here by the endoplasmic reticulum and the Golgi apparatus. But now if we're looking at sequence homology, we can see that, yeah, the insulin from pigs is very, very similar to the insulin in humans and actually can be used in humans. That's called sequence homology. That means that if the proteins are the same, the genes are also going to be the same or similar. And cytochrome C is a very well conserved protein. It doesn't change very much between species, and so it was used to create a phylogenetic tree, which is what figure 3 was. And then we went into how do we get the amino acid sequence? And it's a lot of work. When we get to the part about DNA next week, we'll see that, yeah, DNA is very easy to sequence. Amino acids, very difficult. So the secondary structure is simply alpha helix, beta pleated sheet, and whatever is not is going to just be called random coil. Then I put in a separate place the collagen triple helix. If you look at the word collagen triple helix, the triple helix means it's a trimer. They're already talking about quaternary structure. So that unique secondary structure that exists in the triple helix, I just didn't include it as part of secondary structure because it's unique. If you take apart the quaternary structure of collagen, then that secondary structure kind of disappears. And tertiary structure is simply globular or fibrous. Quaternary structure is simply, is it a monomer or is it not a monomer? An oligomer could be a dimer, trimer, tetramer, whatever. So you're just going to stop me when you have questions, right? And then you should be familiar with the 5 major forces that stabilize protein structure. And I think we did a good job with that. So we went into sickle cell anemia simply because it was an example of how a single amino acid change in one of the subunits of hemoglobin causes hemoglobin to have a different shape. And when you pack it into a red blood cell, it causes the red blood cell to then also have a different shape. And that's kind of an important disease that we should know about. And then you should know the difference between hydrolysis and denaturation. If I denature a protein, that means it lost function. If I denature an enzyme, well, it's not catalyzing the reaction anymore. But that could be loss of secondary, tertiary, or quaternary structure. Primary structure is not lost. So here are some things that denature proteins. And in terms of classification, then we can do it by function. We could do it by composition. Now that we've done chapter 30, notice that I was very careful not to put the word coenzyme in here because it doesn't necessarily mean that it's an enzyme, right? Okay. And then we went into a survey of a bunch of proteins and their functions. So what are you supposed to get out of this? Well, you don't need to remember that glucagon has 29 amino acids. It's just, you know, a fairly small protein that is functional. So hair and fingernails and the other stuff are made out of keratins. Fibroin is really silk. Tubulin, microtubules of the parts of the cytoskeleton.
The first four that you list, are those all simple proteins?
These four over here.
Yeah.
Cytochrome is not. Keratins, probably. Yeah, these are probably, yeah. But cytochrome C is not because it has the iron in the heme group, right?
Okay, thank you.
Mm-hmm. So connective tissue. So these are made out of elastins, collagens, and I mean, this is just for general knowledge, right? You take an anatomy physiology class, they're talking about this stuff, but they're not going to ask you for the exact structure. Nobody would ask you for the exact structure. Yeah. So here's another disease. So vitamin C is required for the cross-linking of collagens. And if you don't have enough vitamin C, you'll get scurvy. Scurvy is a connective tissue disease, right? And then the muscle proteins. I mean, I don't expect you to know the exact details of everything, but just to get a general understanding of what muscle proteins do. So you should know there's a thick filament called myosin, a thin filament made up of mostly actin, but some other proteins in there. There's other proteins that don't get much mention of things. Anyway, so it's a complex but really simple and elegant system how it works. So this is structural. It's a structural protein, right? Muscles hold your body together. But it's also a contractile protein. And notice that when calcium binds to this troponin site over here, it causes a conformational change. And we've talked about that many times, right? And that conformational change, because of calcium going into it, shows that these now myosin binding sites are exposed. So here, the thin filament cannot interact with the thick filament. Once the calcium comes in, yes, it can. So a couple other things, and I showed you some complex pictures, which you'd never be responsible for drawing or anything. And here's another disease, muscular dystrophy. It's where the muscle gets destroyed because it has a defect in the gene for dystrophin. Okay? So dystrophin is a long protein with numerous coils that act like a shock absorber during contraction. This makes you think that I probably should put this part right here next to that. I don't know. That was the whole idea of me putting that there. It's a new concept. It's only been around for a few years. Then we looked carefully at myoglobin and hemoglobin, and we saw that they both do the same thing, is that they bind oxygen with the heme group. Myoglobin is a storage protein, where hemoglobin is a transport protein. So hemoglobin grabs the oxygen from the lungs, takes it to the tissue, and myoglobin that's stationary in the tissue has a stronger affinity for oxygen than does hemoglobin, so it takes it. That helps get the oxygen off of hemoglobin and into the tissues. And hemoglobin is an amazing protein. Not that all proteins aren't amazing, but it can do so many things. So look, here's the amount of dissolved oxygen that you'd find in blood serum. Here is the amount that you have combined with hemoglobin. So most of the blood most of the oxygen in your body happens because of the affinity of hemoglobin to oxygen. It basically sucks it in. So you guys understand this concept about cooperativity? So when hemoglobin is empty, it's a little bit hard to put the oxygen on it. But the gradient at the lungs is so great that you do put oxygen on it. As soon as you get oxygen to bind to one of these subunits, it causes a conformational change in that subunit that then causes conformational changes in the other three subunits. So now they all want to pick up oxygen. So looking at hemoglobin, it's a little bit hard to get it on there, but once you start getting on there, it goes into that mode. Everything just goes on. At the same time, it's a little bit hard to get it off. As soon as you get it over here, where the tissues are, then it will come off. So this sigmoidal area right here is what we mean by cooperativity. Myoglobin has no cooperativity because there's only a monomer. But the whole idea of us talking about cooperativity is that how is this protein responding to the environment to work best to get oxygen to the cells that really need it? So if hemoglobin comes into a tissue where there's about this much oxygen, what's it going to do? Not much. What about over here? What's it going to do? Oh, it's going to start dumping it. But myoglobin won't be able to do that. Hemoglobin will. So that cooperativity is enhanced because of 2,3-diphosphoglycerate. Okay. So 2,3-DPG makes it harder for hemoglobin to convert into the high affinity conformation. So it makes it harder to pick up oxygen, but easier to give it to the tissues. And in tissues that are actively respiring, there's producing a lot of CO2. So what happens is the pH goes down because there's lots of hydrogen ion. That lots of hydrogen ion then can cause hemoglobin to release its oxygen. So the effect is fairly dramatic, right? But again, that's a way for hemoglobin to discover, I'm passing through this environment. Do they need oxygen from me or not? If the pH is a little bit lower, that means that they need it. Okay. So we talked about a few things. Chloride shift, what is the actual gradient that we're dealing with? Why are organisms, aerobic organisms, so good at getting oxygen? Because if you look at the gradient between fresh air and what's in the mitochondria where the oxygen is actually used, it's a very steep gradient. That's the reason. Okay. Glycoproteins. Membrane proteins probably don't need to talk about much. Cytoskeleton. Yeah, we probably should know more about that. I don't really know what they teach in general biology anymore, but most of this stuff was not known when I took general biology. Matter of fact, when I was a graduate student, one of the faculty members here at UH in biochemistry and biophysics discovered dynein. He discovered it. Okay. So is the mitochondria just like bouncing all around the cell? No, it's fixed because of the cytoskeleton. How do you move things around the cell? Well, microtubules do that. They require ATP and energy to do that. So it takes energy to go through meiosis and all that kind of stuff. Should say mitosis. So here's some information about microtubules. There's different kinds of tubulin. Then we talked about antibodies and antigens. Typical antibody structure, you should know a little bit about it, at least this part. You should know a little bit about this. And you should know that, yeah, what do antibodies do? Well, they're in the blood, they're in secretions, they're associated with membranes, and they're not all the same kind. Many of them cause agglutination, right? Some of them are involved in allergies. And you should know what blood complement is. Actually, pretty darn important. So the FC portion of the antibody, people seem to ignore. They only care about the antigen binding sites, but the FC portion is just as important. That's what causes it to be attached to these lymphocytes here. And when it does go through agglutination, look at all the FC regions that's pointing out right there. So if these were to get phagocytosed later on by a macrophage, then how is the macrophage recognizing this? Well, in part because of the FC portion. So here's the concept of complement. Very complex system, isn't it? So here's what a vaccination does. Here's what happens if you get transfused with the wrong blood type. And the antigens on the blood cells, we looked at those real briefly. So digestion of proteins, we do it all day long. About 400 grams a day. It's amazing. So it is very important that you get enough protein in your diet. So some things that can go wrong with proteins, especially with proteins that are long-lived. So racemization, oxidation of arginine. This can happen at any time, incorporation of the wrong amino acid into proteins. So for example, if that did happen, how long will that effect be? It will be as long as that protein is intact. Okay. Which may not be very long, but it may be longer in like a collagen or something. So glycation, that's where you put reactive carbohydrates onto the protein, changes the property of the protein. Chaperones and the ubiquitin proteasome pathway. So chaperones are simply proteins that take other proteins and try to fold them back into its native manner. And so if the protein that's denatured can't be refolded, then the ubiquitination system kicks in and it marks that protein for death. So here's some misfolded proteins. So we, at the expense of ATP, you put ubiquitin proteins on there and you mark it. Now, because ubiquitin is there, the proteasome can bind the ubiquitin thing right here, take that protein and suck it in through the active site and start chewing up the protein. If the protein doesn't have ubiquitin on it, then this thing can't do anything.
So do sickle cells go through that process?
Sure, yeah. Yeah. Problem with people with sickle cell anemia, when they replace their blood cells, they replace them with more sickle cells. So that's the problem. If you were to transfuse somebody with sickle cells, then the sickle cells would last a couple of days and then totally be degraded, and there'd be no long-lasting effect. And then prions cause some brain degenerative diseases. And a prion is acting like a chaperone, but a bad one, an evil chaperone. So it takes the regular prion protein, which does have a biological function, right? And it converts it into the beta form. Mostly alpha structure, now it's mostly beta structure. And then this then acts like an enzyme. Now it's not truly an enzyme, all it does is change the folding of a protein. So it's more like a chaperone. Okay, and then beta amyloids over here. So here is right where that protein comes from that forms the beta amyloid. And yeah, now this they can't be chewed up by the ubiquitin system or the chaperone system or anything else. It's just, what do you do with this? And does it cause problems? Yes, it does. Of course, now they know that in Alzheimer's progresses regardless of the amount of amyloids that's in the tissue. So people with amyloids in the tissue don't necessarily show symptoms of Alzheimer's, and people with Alzheimer's don't show oftentimes tremendous amounts of beta amyloid. So it's not a direct proportion relationship. And so now they're looking at other factors, of course. So we looked at the immune system, how it tries to protect you from bad proteins. So you don't really, this is not an immunology class, so I'm not going to ask you, well, what do B cells do or what do T cells do? But you know that they're related to the immune system. So I'm doing a kind of a drastic rewrite of the next couple of chapters. And somewhere along there, I got to put in in one of the chapters, a thing about inflammation. I have inflammation kind of scattered all over the place, but inflammation is really a problem. Look at what's going on with people who have SARS-CoV, acute respiratory syndrome. It's really because their regulatory T cells aren't doing the job, so the cytotoxic T cells are just wreaking havoc on the patient's own lung cells. So it's meant to help protect you, but when it goes wrong, it really goes wrong. It really becomes an autoimmune disease. So if you think about allergies and things like that, it's your body's way of telling you, wherever you are, get out of there. Whatever you're smelling, whatever you're eating, stop doing that. And so if people have allergies and they remove the allergen, then the symptoms go away. Not right away, because you have all this immune response and all this other stuff that has to subside. And then, you know, in modern days, children are protected by their parents. They don't want them to suffer anything. And I went through that raising my own kids, because their mother was basically trying to, you know, protect them from everything. And I'm like, well, no, they got to be able to live in the real world. Today's modern hygiene, you can basically keep your kids in a bubble, but then they're not getting exposed to all the antigens that they should be getting exposed to. So they're not making, you know, the ability to how do you say, respond to the environment correctly. It's not a good thing. So that is a really good comparison between the Amish and the Hutterites, because it really shows you that similar genetics and different lifestyles can make a difference with allergies. And then we talked about gluten and celiac disease. So here's gluten right here. It's made up of gliadin and made up of lutein. And this does not get completely digested. That's the problem. So again, if it was just gluten going through your intestines, would it be a problem for your digestive tract? And the answer is probably not. Is it good? Maybe not. But what makes it worse is when your body mounts an immune attack against it. So these damaged cells release all kinds of cytokines and all kind of inflammation factors and everything else, and that mounts the immune response. And then these yeah, the immune response is really what causes the problems. So I'm simply calling it an autoimmune disease. So in here somewhere, I should probably put how many of your cells are involved in digestion and getting nutrients. I have the number somewhere. I don't remember what it is, but it's a tremendous number. Look how long your intestines are. So if you go look at rudimentary organisms, like a good example is a sea cucumber. Sea cucumber is almost entirely guts. There's nothing else to it. So we need a tremendous amount of our body to be involved in getting nutrients for us. And if that becomes damaged and we don't get nutrients, we don't stay around for that long. That's why anything that attacks your digestive system is actually very dangerous. Okay, so that basically takes care of that chapter. Anybody have any particular questions on this chapter?
Is there a way to control the overactive cytotoxic T cells that you talked about?
Well, I mean, people are trying to figure out how to do that. So it's the T regulatory cells that we need to actually think about. And so there are immunosuppressive drugs that could be useful to some people. And I think that they are finally starting to use those in SARS-CoV patients, because that's part of the problem there. Like a lot of the biochemists, we recognize that right away, but we didn't know exactly what was going on with the COVID stuff, so we didn't know if that was going to work or not. But they tried it, and they do have some success by giving immunosuppressive drugs there. But then what we're also finding out with the SARS-CoV patients is that they're getting all kinds of secondary infections. So people who have recovered from being in intensive care, they come out and they're not recovered because now they have all these secondary infections. And many of those secondary infections were derived in the hospital, and they are penicillin-resistant drugs. I mean, penicillin-resistant organisms. So that's another issue. So when you just start going around using immunosuppressive drugs for something, you got to be really careful about it, because your immune system is now compromised. So to prevent this, it's a fine line. Okay, well, I go to the next chapter. Got so many windows open. Earlier today, I'm on this committee to figure out how to get more out of La Lima. And so we're all on Zoom, and we're all trying to show each other stuff, and everybody was having problems, like so many windows open, you can't find the window you're looking for. It's kind of funny. So anyway, enzymes. So We talk about the holozyme. It's the whole enzyme. You can break that down into the apoenzyme, which is simply the protein part. And because most proteins are conjugated, so I should say most enzymes are conjugated proteins. So the cofactor can be called a coenzyme. It can be called the metal ion. It could be a prosthetic group, or they can name it by what it is, like a porphyrin ring or something. But the difference between a coenzyme and a prosthetic group is this prosthetic group is covalently bonded to the protein. So there will be something on the exam about recognizing the 6 classes of enzymes, but not by number. It'd be like a matching thing or something, right? Like, here's a reaction. Which category is it in, right? So if you ever see a redox agent, whether it's in the oxidized form or the reduced form, automatically it's going to be an oxidoreductase. And I think we've seen enough organic chemistry, we could also recognize that. So the difference between pyruvate and lactic acid is what? Oh, that alpha keto group right there got reduced into an alcohol. If I reduce a ketone, I get a secondary alcohol. If I oxidize a primary alcohol, I get an aldehyde. And a transferase is simply you transfer a group of atoms. It could even be a single atom, like a chlorine or something like that. You transfer it from one substrate to another. Okay. So here's an example of a phosphotransferase, right, from ATP to creatine. Here's an example of a methyltransferase from this donor molecule to cytosine. That's how you get methylated DNA. A hydrolase uses water to break a bond, but don't forget, these are also the same category if they're going in reverse. So here I have water on one side, here I have water on the other side. That's also a hydrolysis, isn't it? Or excuse me, a hydrolase. And lyases is usually group elimination, and you form a double bond. That's the usual case. That's the only one I'll harass you with. Okay. So I'm looking at this. Is this a hydrolase? Is water splitting something over here? And the answer is no. Well, what's the water doing? It's getting added to that double bond right there. Oh, okay, so it's a lyase. Or I'm looking in the opposite direction. Where'd that water molecule come from? Well, it came from either here or here. I can see it came from here, directed by an enzyme, right? So going this way, I formed the double bond. Going that way, I lost the double bond. So it's a lyase. And isomerases convert one isomer into another. So here we broke a bond and reformed the bond in a different place. So we turned galactose into glucose. Here we took the phosphate group on the number 2 carbon and moved it to the 3 carbon. Those are isomers. Then ligases is the one that involves large amounts of energy. So you will see lots of triphosphates involved in this. Okay. So notice that all these over here are two directional. And I put these as one directional because they're so exothermic that they're unlikely to go in the opposite direction. Okay. And many of these actually are driven towards completion by Le Chatelier type principles. So for example, if you look at pyrophosphate, well, what happens to pyrophosphate? That's still a phosphoanhydride bond there. Well, it's going to be hydrolyzed. So there's an enzyme that will hydrolyze that into phosphate. But phosphate is not involved in this equilibrium. So every time you remove the pyrophosphate, you're removing one of the products. The result is you're taking that away, you're going to shift the reaction more in that direction.
So if we were to see that second example on ligases, serine plus alanine, would we know that that was a ligase because of the arrow is going one way and it's not the hydrolase?
Well, in that particular case, you would have to know that to make the peptide bonds with the ribosome requires the hydrolysis of GTP. So I know that because I read the chapter that's coming up.
Is there a way for you to show us how that gets drawn?
So somebody who was just talking, I could not hear that. They were very, very low.
Is there a way that you could draw that to show us?
Right here?
Yes.
Not really. I have to go through the whole rigmarole in the next couple of chapters.
Okay, so is that something that's going to be on the exam?
No, I'm not going to put something in there to trick you.
Okay.
One of the reasons why I chose these two examples right here is because people have taken biochemistry, and even biochemists, they don't think of these as being a ligase when they actually are. But anyway. So then we should know what complete and group and linkage stereospecificity means. And I was trying at this point, trying to get you to start thinking about what I call the conservation of genes. The more proteins you have, the more genes you need to produce them. The more genes you have, the more things can go wrong because you can only have so much DNA in a cell, and after that, you can't control it. It's just too complex. So then you should know the difference between the lock and key theory and the induced fit theory. And you should have some idea about what makes an enzyme work. So when we get to the point of talking about DNA, one of the things I'll emphasize to you is this. There isn't very much information in the linear piece of DNA. Hardly any information at all. But if you look at what an enzyme can do, for example, there's so much information involved in what that protein can do. Where is that amino acid? Where is this amino acid? Where's that functional group? Where's that cofactor? How does it rotate a single bond here when it binds to its substrate? The amount of information in the protein is tremendous. But most people don't get that because they think, oh, DNA is simply linear. But no, tremendous amount of information in a protein. And information is the same thing as energy. So why does the enzyme be able to do what it does? Because there's so much energy involved in the production of this protein. And part of that makes it specifically bind strongly to this particular substrate. So much so that if you take them apart, you're creating potential energy. So when you let them bind, that potential energy gets converted into what? Kinetic energy, which is a form of binding energy. It causes all the conformational changes in the protein, all the conformational changes in the substrate. And when you do that, you put stress on the bonds to be broken. So again, a catalyst can do nothing more than lower the energy of activation. Okay. So here's another way of looking at how the enzymes lower the activation energy. They bind the reactants and bring them into close proximity. So if you think about why they bind reactants and bring them into close proximity, it's because there's so much energy in the design of this. That's why. Regardless, it increases the concentrations at the active site, which increases the rate. Then it puts the reactants into the correct orientation so that all the collisions will be fruitful. And it puts strain on the bonds to be broken. And that's what I meant by this binding energy right here. And then finally, they provide functional groups. So the functional groups can be acidic, they can be basic, they could be a metal ion that can supply, either was donate electrons or accept electrons for a temporary situation, right? Cofactors. So then I went into great depth at this reaction mechanism, knowing perfectly well that I'm never going to ask you anything about it. But once you see how it works, I think from now on you will remember that, oh yeah, enzymes find a way that we can totally understand how they catalyze their reactions. Totally changes the reaction mechanism. I think you can also understand, well, why do some enzymes have iron atoms and other cofactors like that? Because they're directly involved in changing the reaction mechanism. That's why. So ultimately, an uncatalyzed reaction would have to do this. They would have to bang into each other hard enough that might require increased temperature. They would have to bang into each other in the exact right orientation, which is not going to be most collisions. So it'd be pretty difficult to do. So maybe here's the binding, putting strain on the first part, right? Here's where iron gets involved. Now it makes the lower energy complex with the iron. See, you break it up into these individual small steps, and the activation for each of those small steps is not very big. So it's easier to accomplish. So the reaction rate, you should know what a rate is. So I was very surprised when I found out that we had graduate students that took biochemistry and got A's and this and that, and they don't even know what a rate is. It's like, come on. And then I'm not bothering you with the Michaelis-Menten reaction stuff. I want you simply to understand the concepts. So here I took one rate. I took this rate at a given concentration of substrate. If I did it at a higher concentration of substrate, I'd get a faster initial rate. And I would get a lower rate if I did it at a lower concentration. So I take the slopes of all those and I plot the rate against what substrate concentration I was using. And I get this kind of a curve right here. Okay. So the two things that are important to describe a hyperbola, if the hyperbola goes like this, right, is where does it asymptote to? Where does it get closer and closer to, but never really mathematically getting there? You need to establish that data point. And then once you have that established, you need also another data point to say, does it go close and then drop quick? Or does it like start dropping like this and then go like that? So the other place is you take half of the Vmax and you hook this in right there. So that locks in that point. So if I got that data point there and that data point there, I can describe that hyperbola. So the two important pieces of data are what is the Vmax, the maximum rate you can get for a given amount of enzyme, and how much substrate do I have to give to get half of that rate? That's what the Km is. So what does it mean if you find an enzyme that has a very large Vmax? Well, the enzyme works very rapidly. What does it mean if the enzyme has a small Km? It means that, it means that you don't need a lot of substrate to make it go that fast. And then an analogy to the binding of a drug to its receptor protein. Okay, what does it mean if you have a very high binding? Well, that means that there's strong binding between that drug and the receptor protein. What do we call the KB? Well, I can get half bound with a small amount of drug. That means it's a very strong binding. That's what it means. So if I got two drugs that bind to the same receptor, one the KB is right here and the other the KB is right there. When I put them both into the container with that receptor, which is the drug that's going to bind first in most of the receptors? It's going to be the one with the smallest KB. Similar with enzymes in the Km. So say I invented an enzyme competitor, its Km was like right here, and the Km for the regular substrate is out here. Well, that means that the inhibitor I just came up with can be a strong inhibitor, strong competitive inhibitor of the natural substrate. So factors that affect protein stability that we talked about in the previous chapter, right? So anything that affects protein stability is also going to affect the rate of the enzyme. So if I'm looking at this one right here, the initial rate is low over here. When I start to increase the pH, oh, the enzyme gets faster and faster and faster. But then it starts to slow down. So where is its maximum? Oh, it's about blood pH, so this is probably meant to be active in the blood. And then effector molecules. So we talked about inhibitors for the most part. So we have irreversible inhibitors, we have reversible inhibitors. You know, the reversible ones are either going to be competitive or non-competitive. So here's a competitive inhibitor. They're competing for that same binding site. If the Km for binding, the competitive inhibitor and the substrate were the same, then they'd be equal in competition, right? And then you have the non-competitive inhibition, where there's no competitive inhibition because they're not competing for the same site. So it's an allosteric inhibitor because it binds to a different site. Okay. So in other words, say the non-competitive inhibitor has a small Km or Kb for the enzyme. It's going to bind, and it's probably going to prevent the substrate from getting in there. No matter how much substrate I got, I can't overcome its inhibition. Over here, the more substrate I put compared to the amount of competitive inhibitor, the more I'm going to outcompete the competitive inhibitor, and my rate will go up. And the other type is really just a mixture of the other two. So to show that, I just kind of moved the binding site over so they share some characteristics of the same binding site, but then they don't share other characteristics of the same binding site. Okay. So questions so far? So I'm sure you've heard of the microbiome. The reason why you've heard about it is because as soon as something comes up in science, the pharmaceuticals, the nutraceuticals, those businesses are just all over it. Because it is a billion-dollar industry. They lobby Congress. They prevent laws from being passed to protect the public. It's so much money involved that, yeah, it's ridiculous. But right now, I do not know of any probiotic that has been proved to be effective for the general public. But there's a tremendous amount of research going on in it by earnest and legitimate researchers. There's also a whole bunch of other research going on where these people that sell this stuff are trying to find people to use science to pretend like it does. So anyway. So anyway, antimicrobials are against anything that's microbial. But they could be bactericidal, they could be bacteriostatic. So antimetabolites are generally bacteriostatic. But what that means is it gives your body enough time to come up with another defense against it, and you can survive the infection. So you can see that sulfanilamide is a competitive inhibitor to paraminobenzoic acid for the enzyme. This is also a competitive inhibitor. Then talking about antibiotics, then you see this is the beta-lactam ring is simply a highly stressed ring. So when it does bind to an enzyme, it can easily break that bond open and form bonds with the enzyme itself. But bacterial-resistant, I should say, not bacteria-resistant bacteria, drug-resistant bacteria can get the enzyme called beta-lactamase, or penicillinase is another word for it, that can break that ring open. And then it's useless, it can't do anything. So here are some of the things that they're trying to replace antibiotics. So a virus that attacks bacteria. I remember when I first learned about that, it's like, what? A virus that attacks bacteria? Revenge. So if you look at penicillin, what it does is prevents the bacteria from building its cell wall. Okay. If you look at bacteriophages, what they seem to be doing for the most part is they're also attacking the bacterial cell wall. Not all of them. And that's how they found lysins. So lysins are enzymes that break down the bacterial cell wall. So it's pretty unlikely that a bacteria is going to be resistant to that actual enzyme right there. And bacterial sins are toxins that are not antibiotics. And then cationic peptides are basically used to break up the biofilm. And then I'm just going into some of the things that they're trying to do to find the new antibiotics. People are taking everything they can to come up with new antibiotics. By the way, I saw this thing on a TV special. I forget, it was 60 Minutes or something like that. And they're interviewing the CEOs of these companies that were involved in making antibiotics. And they basically are getting ready to file Chapter 11 bankruptcy because people aren't going to the hospitals and doctors as much because of the virus. So doctors are not selling as much antibiotic. And they've had to shut down their research labs because they can't afford to go look for new drugs. Pretty scary. So how does the cell regulate enzyme activity? Well, one of them is simple product inhibition. The product itself inhibits in a competitive manner the interaction between A and the enzyme. So for example, if B is not being used up, its concentration goes up, and it's going to become a competitive inhibitor for A. Then feedback inhibition would be not the immediate product, but some product further down the line. It could even come from a completely different pathway. Regardless, it binds to an allosteric site over here and it inhibits the enzyme. And then we have all kinds of allosteric effectors. And allosteric effectors could be exactly what we're talking about here with feedback inhibition. But these effectors are not called normally feedback inhibition because they can also be activators. So for example, ADP, high concentrations of ADP could activate one group of enzymes, but at the same time inhibit another group of enzymes. And when we get into the chapters on metabolism, we'll find out that this is simply logical and there's nothing illogical in metabolism. And the easiest way to see that is this. When do you do biosynthesis? When do you start to go produce more DNA and go through cell division, all this kind of stuff? Is it during a time of starvation? You don't have food, you don't have energy. Now's the time you're going to use energy to biosynthesize something. When do you do biosynthesis? When you have adequate energy. If you have adequate energy, that means you got mostly ATP. You don't have a whole bunch of ADP. See how that works? And it makes no sense to be degrading something for energy and at the same time building up that same thing in biosynthesis. Makes no sense. So covalent modification or hormone regulation, and we go into this and look at other systems of this in chapter 31, the next chapter in unit 4. But here's a very simple one. So it involves acetylcholine, or adenylate cyclase, I should say, acetylcholine. Anyway, so here's where a hormone binds to its receptor, and that hormone then causes something to happen in the cell. It doesn't cause it directly. This is the first messenger. In this case, cyclic AMP is the second messenger. So cyclic AMP came from inside the cell, from ATP. The hormone doesn't even have to come into the cell, but its effect happens in the cell. So in the next chapter, we'll be looking at, well, what happens if I take cyclic AMP and I were to simply inject it into, say, a liver cell? I'm going to get the same effect on that liver cell without the hormone. See what I'm getting at? So nothing's going to happen until the hormone binds here. But if I took a little needle and I injected cyclic AMP directly into the cell, I'm going to get that physiological response. Okay. So you might want to start getting yourself familiar with this sequence of events here. Okay. How come I have 2 5s over here? Better change that. So here's kind of an elegant system. So here's an enzyme that is turned off. This enzyme is not functional. These are the catalytic groups that would be the active enzyme right here. But these regulatory subunits prevent it from acting as an enzyme. So this right here is turned off. How you turn it on? Cyclic AMP has to get into the cell somehow. If I were to inject it directly with a needle, or whether the hormone binding to its receptor protein caused it to be formed by adenylate cyclase, it doesn't matter. Once enough cyclic AMP is in there, it causes a conformational change in the regulatory subunit, the thing falls apart, and now we get the active enzyme. Okay. So what does the active enzyme do? Many of these are phosphotransferases. So it takes ATP and it takes the protein, puts the phosphate onto the protein, it modifies the protein, and that either turns the protein on or turns the protein off. And that's what causes the change in the metabolic state of the cell. Okay. This enzyme being turned on or enzyme being turned off. Make sense? So part B has to do with this. Do I want the cyclic AMP to stay in this cell forever? Notice that I'm not synthesizing new protein over here. I'm just turning the protein on. I'm activating it. If you think about the fight or flight response, how long does that last again? Like a month, a week, a day, an hour? No, it lasts for a couple of minutes and then the effect is over. So all these things have to be short responses to the environment. They're not like a nerve cell. It's not meant to be just turned on forever. It's like this, it's over with. So there has to be a way to get rid of the cyclic AMP. So the cyclic AMP activated this enzyme. In the same cell, phosphodiesterase is there, not doing anything. How does it get turned on? It gets phosphorylated by these same enzymes over here. And what does it do? It takes cyclic AMP and breaks it down, removing it. So this B system right here is to make this whole thing short-lived. Okay. Let's see. 1, 2, 3, 4, 5, 6. How come I'm saying this is 6? I don't know. Later on I have to look to make sure that makes sense. So questions about that? Figure 3. So let's kind of review this. I want to connect the two figures together. So some stimulus from the environment causes the release of the hormone. It travels through the blood, sometimes through the lymph. It won't be able to do anything until it finds a cell with a receptor protein on it that binds to that particular hormone. So it binds the receptor on this cell, this cell type. Conformational change out here causes a conformational change over here. Notice this is an old picture. I had to put the G protein in there. And this is adenylate cyclase, gets turned on. Okay. So what does cyclic AMP do? Well, it takes this protein kinase right here and activates it. We saw by taking off those regulatory subunits. And what does protein kinase do? Protein kinase is a phosphotransferase. Now that it's active, it simply goes anywhere it finds a protein and puts a phosphate group on it. If you are a liver cell, one of the enzymes that you have a lot of in your cell is glycogen phosphorylase. So that will become a target for the phosphotransferase. Put the phosphate on here, now it's active. What does this enzyme do? It breaks glycogen down to glucose, and then you can release it to the bloodstream, and then you get superhuman powers. For a little bit. And I just realized that I wasn't sharing the screen there. Sorry, I guess I have to do that over again. Is it sharing now?
Yes, and that's okay. The more repetition, the better.
Okay, so I'll do it over again. So here's the stimulus from the environment. Causes a release of the first messenger, which is the hormone. And the hormone is only going to be bound to cells that have the receptor for it. So when the receptor binds that hormone, it causes a conformational change. It activates G protein, which then activates adenylate cyclase, which then takes ATP and converts it into cyclic AMP, which is now the second messenger. So at this point, I'm going to get this response in the cell. Even if I were to not even have a hormone, if I were just to simply inject a cyclic AMP directly, I would still get the same response. So the wishes of the hormone are carried out by cyclic AMP now. That turns on protein kinase, which is a phosphotransferase. So what does a phosphotransferase do? It simply puts phosphate on any protein it runs into. But in a liver cell, one of the major proteins that you're going to run into is going to be glycogen phosphorylase. So you put a phosphate on here, and you've activated glycogen phosphorylase. And that's the enzyme that breaks glycogen down into glucose, and then glucose gets released into the blood. So what happens if I have a liver cell and I inject cyclic AMP into it? It's going to release glucose. What happens if I have, what's another cell? Say a kidney cell, and I inject it with cyclic AMP. It's not going to release glucose, but it might release potassium or uptake potassium or something like that. Anyway. So then we also have isozymes. I think I did a pretty good job with this diagram here, explaining that. So the same protein or the same enzyme in one tissue catalyzes the same reaction in another tissue, but they can be a little bit different. And then zymogen activation. You take these proteolytic enzymes over here, it would be pretty dangerous for you to be a cell, and your job is to hold these proteolytic enzymes. Because if they were to get loose inside you, they're going to eat you. So that's why they're stored in inactive forms. Same thing with this over here. And then finally is simply separate the substrate from the enzyme, physically separate them. So now you control the reaction rate by this membrane protein right here, this transport protein. So there's multiple ways to describe how a molecule is transported across the membrane. So here's the basic categories, and then those categories can be further described by how they work, right? So I tried to illustrate them all here. Okay. So requires no energy, requires no energy, requires energy. If you use energy, you can go against the gradient. You're not just stuck on diffusion. You can concentrate different things. Then I went into a little bit of detail about the ABC binding cassette, or excuse me, ATP binding cassette proteins. Not that you really have to understand exactly. This is artist concept, by the way, based upon the data. But it gives you an idea of how they can work. So because it uses ATP, it's not passive. It can go up a gradient. You can concentrate things. So I guess we kind of covered everything. So any other questions about anything?
For the inhibitors, the competitive and non-competitive, how is the non-competitive inhibitor reversed if it's considered irreversible?
Oh, well, they can just diffuse away. So it's Le Chatelier's principle always, right? Because it's not a covalent bond. Where was that? It's way up here.
On page 8, I think.
8, okay. Yeah. So all these inhibitors are subject to equilibrium, okay? Because they're not permanently. If they were permanently attached to the enzyme, then they would be irreversible inhibitors. So say this non-competitive inhibitor over here happened to be a molecule of ADP. So how is the concentration of ADP in the cell regulated? How is it changed? Well, if the cell has a whole bunch of energy and it starts converting all of its ADP into ATP, then this eventually is going to fall off and get converted into ATP. So that inhibition then just goes away. If you don't have this, then it's not going to be an inhibitor.
It's kind of based on the concentration of the substrate.
Exactly. And the concentrations of all these things in a normal active cell are just fluctuating all the time. Yeah. So if the concentration of the non-competitive inhibitor decreases, well, then the enzyme will be active again. Yep, nothing is forever in a cell. Constantly fluxing.
Got it. Thank you.
Sure. Okay, so anything else? Okay, well, I will put this up onto the YouTube site and you'll have that. And if you want to take the exam tomorrow, just make sure you email me and I'll put it into your Dropbox. Sound good? Okay, I know it doesn't sound. I know. Okay, well, you folks have a good day, okay?
You too.
Thank you. You too.
Here, bye.
Bye.